Assalamualaikum and good day everyone. So we will continue with antibody labeling. Okay, antibody, if we just simply try to observe the antigen and antibody complex, if they form in a large, huge quantity, that we can observe them in the form of agglutination or precipitation. But if the reaction only happen in a very little amount, or maybe single reaction between one antigen and one antibody, there is no way that we can detect them using a normal method of observation by naked eye or microscopically. So we need to put label on the antibody, so that label can be used as a beacon or will highlight the reaction between the antigen and antibody. So this antibody can be we can observe here the critical components of an immunoassay, where we have analyte. This is the thing that we want to test, the thing that we are looking for. We call them as commonly used in the hospital or in the diagnostic lab as analyte. This analyte can be pathogens, antigens, protein, or any other substance that can bound with the antibody that we are using. Specific antibody, so specific antibody for that specific analyte that we are testing, and also detectable label. So this is the most important part in immunoassay. This is the thing that will give color to the reaction. We give light in form of radiation, so many type of signals that can be used to measure later. Okay, first we can attach small molecules to the antibody. We can attach particles to the antibody, or we can attach the antibody to a particle. We can use isotope, enzymatic protein, enzyme, and also fluorescent dyes. So what type of signal that we are looking for in this different label? First, by using small molecules, the signal that we are looking for in the form of formation of color, fluorescent light, for particle also color in fluorescent light. Isotope, we will measure the radioactivity or the radio emission level, because isotope is an radioactive signal label. In enzyme, the signal will be in the formation of color or light from reaction of chemiluminescence. In fluorescent, we will looking for also light in the form of fluorescent light. So all these signal can be detected by specific analyzers, specific machines, and some can even be detected by our naked eye. For example, color reaction in the strip test for pregnancy test, we can observe the formation of a red line on the immunoassay. First, we look on this label using small molecules. One of the most popular is biotin. Biotin can be used in lateral flow immunoassay, Western blot, ELISA, flow cytometry, immunofluorescence, and also immunohistochemistry method. Do check out briefly on this type of immunoassays to get the idea how they work. And also nanoparticles. Nanoparticles is getting more and more popular nowadays in the research and also diagnostic field. We can use gold nanoparticles, silver nanoparticles, lateral flow immunoassays, Western blot, and ELISA, to name a few. So now we look an example by using nanoparticles. How these small molecules or nanoparticles attached to antibody can help in giving signal in immunoassay. Okay, we begin with the antigen-specific antibodies. So these antibodies is antiserum for a specific antigen that we are looking for. So imagine that we are testing for COVID-19 virus in a patient sample. That means the antigen should be coming from the COVID-19 virus itself, SARS-CoV-2. So we already produce the antiserum to it. So this antiserum will be labeled with gold nanoparticles. So in the form of nanoparticles, gold can be observed in the form of red color. And then we will mix this antiserum labeled with nanoparticle with the patient sample. And the patient if the patient sample contain the target antigen that we are looking for, of course they will bind, the antigen will bind with the label antibody complex. And this complex will be passed through a line of capture antibody, and this capture antibody also specific to the target antigen. So we have two types of antigen looking for the same thing. First, the antigen that carrying the labels here, also specific to the target antigen, and also second, capture antibody also looking for the target antigen here. So this complex will pass through the line of capture antibody, and they will be captured, hence the name capture antibody. So the capture antibody will bind to the antigen on the antigen antibody complex, like a sandwich here. So this binding will form accumulation of large numbers of gold nanoparticles. So eventually, if their amount is large enough, they can be observed with the naked eye or with the analyzers. So we can found the application in the rapid strip test. So this rapid strip test is very common. They are applied in diagnosis of course COVID-19, dengue, hepatitis B, many types of tests for drugs, for illegal drugs, where we put urine here and antibody will capture the illegal drug and so on. The process will be the same. We just change the specificity of the antibody. And also we can found in even in 7-Eleven, you can see them, the pregnancy test kit. So the red line here that we interpret as positive. Positive mean because we can understand here, looking how the red line is formed. Actually, it's the accumulation of gold nanoparticles, and when the numbers is large enough, they can be observed. The second, particles in the form of microsphere, small balls, we can say, microscopic balls. they can be made from polystyrene, latex based polystyrene, fluorescence or magnetic. So this is the appearance of the microspheres. They are really small, microscopic in size, even nano in size. This is a surface electron microscope image. So these microspheres can be used in lateral flow immunoassays, latex agglutination tests, fluorescent microsphere immunoassays, and magnetic ELISA also in magnetic immunoassays. So again, do check out brief information on this type of immunoassays. For fluorescent microsphere immunoassays, the Luminex is the name of a company that first developed this technology. So they are very popular. They are the only company that provide this type of immunoassays. First, we have a microsphere attached with the specific antibody. And once the test microsphere that contain the specific antibody mixed with the sample, if the sample contain the target antigen, it will bound to the antibody on the microsphere. So this provide identification of the antibody, because the microsphere is usually being used to as an identification the way of identifying the type of antibody that we are using. As for signal, so we will detect the signal by putting a second antibody. So this is the detection antibody. The detection antibody can be labeled with anything. For example, here we put a fluorescent reporter as the label. So this fluorophore or fluorescent reporter can be detected later by using the Luminex analyzer. So now in this reaction, we know the type of antibody. Since we know the type of antibody, type of microsphere, we know the type of antigen that it target, and then we know that it is positive because we can detect the signal here. And then the microsphere that already tested or react with the patient sample will pass through the Luminex analyzer. So green laser will be used to count either it is a positive or negative, because if without this specific antibody with the label, there will be no fluorescent signal detected by the green laser, so it means negative. And then the red laser will identify the type of bit that passing through. If we are looking at this type of reaction, positive, where they have the target antigen in the sample, the analyzer will identify the bit and then will detect whether test is positive or negative. If there is no target antigen on the sample, the bit will still go through the analyzer, but there will be no signal coming from the fluorescent reporter, so the test will be negative. So this way we can count the amount of target antigen available. So do check out the principle of the Luminex analyzer. Another example, common use of microsphere is in the latex agglutination test. Here in circle A shows a positive test because we can observe agglutination of the latex particle, and the other circles negative because no agglutination. The agglutination actually form when the antibody attached with the latex microsphere agglutinate with the antigen that we are looking for. So the baby looks like a curdled milk. So this test can be interpreted by the user. We don't need to use any instrument. Second, by using isotope. Isotope is a radioisotopes label that can be put on the antibody or even on the antigen. So in all the labeling process, they are not specific to antibody. We can reverse the reaction, just like we know that serology we can detect the antigen or antibody. So if we reverse it, we put the label on the antigen. In this case of example I am showing here, and the signal radio emission will be detected using radio immunoassay instrument. Radio immunoassay using isotope is the most sensitive. They are the best method in identifying antigen or antibody, and they are very specific method of in vitro assay techniques, and they can be used to measure the concentration of antigens. And we can measure how much, if the antigen is a virus, how many virus is there in the patient blood, compared to most tests like for example for the latex agglutination test. We cannot determine how many the number of antigen in the sample. We can only know that the patient is positive or negative for that particular analyte that we are looking for. But by using isotope, we can measure the amount of antigens or antibody. Example, in radioisotopes label target antigen. Here we label this antigen is a reagent antigen that being artificially produced in the lab. So and then they are labeled with the radioisotope. So this is radioactive material that will give signal later, plus sample unknown concentration of unlabeled target antigen. So of course we will test with the sample. The sample might contain the same antigen, but they are not labeled because they are coming from the patient serum, and their concentration is unknown. These two antigen later will compete in binding with the specific antibodies or antiserum that we will be using in the radioimmunoassay. Okay, first we look on the control part. Control mean we are using radio label antigen. There are no patient sample involved in this control testing. We just using all the reagents, the reagent of radio label antigen plus the antibody reagent of the antiserum. So first, the antigen reagent that is already labeled with the radioisotope will we mix with the antiserum specific to the antigen and they will form antigen antibody complex, and afterward we will measure the level of radio emission. So in the previous method, like in nanoparticles, we can observe the formation of light, but in this case they will transmit radio emission or radioactive signal coming from the radioisotope. So we will measure the concentration, and we will jot down the levels here. So control, so this means 100% bound to the antibody, and how much is the radioactive levels is being detected. Then we will proceed with the patient sample. So we assume that the patient sample contain the same antigen, the same virus or pathogen or whatever analytes that we're looking for in the patient sample. So they mean a positive sample, and we will mix together with the antigen reagent that we have in the lab, the radio label antigen that already labeled with the radioisotope, and then they will go through the same process. They will compete with each other in binding with the antiserum. We can observe here some bind to the unlabeled antigen from the patient, and the rest will bound to the radio label antigen reagent that we have earlier. And then we will measure the level of reactivity. For example, we get the level here close to half of the control, so we know that the patient sample contain 50% of the antigen that we are looking for compared to the known level. For example, if this level is 100 milligram or the titer is 100, so we know that this is 50. So this is how we count the concentration of the antigen, by comparing the known level with the level from the sample. Okay, now we go to the using of enzyme as a label. Uses an enzyme that is attached to an antibody, and this can be used to detect antibody or antigen, the same for all the immunoassays or serology tests. The antibody attached with enzyme, and now it is known as enzyme conjugated antibody or enzyme label antibody. To use enzyme label antibodies, samples are incubated with a substrate. Okay, the substrate will be catalyzed by the enzyme to produce a colored product. Maybe they will react enzyme with the substrate, a chemical reaction happen, they will produce a colored product or light. This enzymatic protein labeling is very popular in immunoassays. In color production, they are used in ELISAs, lateral flow immunoassay, Western blots and immunoassays, etc. While light production commonly used in Western blot and also in many analyzers in the diagnostic lab in the form of chemiluminescent immunoassays. So first, we briefly look on how the enzyme link immunosorbent assay or ELISA used to detect the antigen. Okay, we will look from the beginning. Okay, first, patient antigen will be allowed to bind to the well. So this is positive and negative. So we are looking for the red antigen, okay? And enzyme label antibody will be added, contain enzyme, and then rinse. The negative will not bound. After that, substrate will be added, and reaction between enzyme and substrate will produce a color reaction for positive and no reaction at all in the negative, because this patient do not contain the red antigen that we are looking for. Second, in order to detect antibody using ELISA, we will wait from the beginning. Okay, first we will put the antigen that we have, the rate we are looking for. We put the patient sample, they contain the antibody. If they is positive, the antibody, specific antibody will bound to the antigen, and then we will put the second antibody that contain the enzyme that will bound to the antibody that we are looking for, and then we will add substrate. The reaction between enzyme and substrate will produce a color changes compared to sample B negative, because this patient do not contain the antibody in the beginning. In detecting antibody, we are using antibody against antibody. And fourth, fluorescent dyes or fluorophore as a method of labeling the antibody. So the use of fluorophore labeled antibodies, since fluorescent dyes are directly conjugated to the antibody, no enzymes or substrate or binding interaction are required for detection. So in this case, the antibody will remain active, because we will only tag the antibody with a fluorophore. So the fluorophore do not need any chemical reaction to produce light. They only need a UV or laser transmitted to them. Once a UV light or specific laser transmitted to the fluorophore will absorb certain wavelength from the UV or the laser, and then emit a different kind of light with the different wavelength. So this light produced from the fluorophore will be analyzed. Therefore, the amount of fluorescent signal detected is directly proportional to the amount of target protein in the sample. So also this method can be used to quantitate the amount of target protein, antigen or analyte that we are testing. Fluorescent labels are used in flow cytometry, immunofluorescent assay, and fluorescent western assay, to name a few. So this method is also one of the most widely used in the diagnostic laboratory. Okay guys, thank you very much. Assalamualaikum and have a nice day.